Within the silent electrical pulses of your thoughts exists a complex biological landscape defined by the male and female brain. This hidden world functions with a quiet precision. Every human brain contains roughly 86 billion neurons that form a dense web of communication through quadrillions of synaptic connections. If you find this exploration of neuroscience helpful for your rest, consider subscribing to join our evening sleepy science. Modern neuroscience increasingly views sexual dimorphism not as a collection of rigid opposites, but as a vibrant mosaic of overlapping traits. While physical differences exist, the vast majority of brain structures are shared across all humans, creating a baseline of universal cognitive ability. Researchers at institutions like the University of Oxford have observed that individual variation often outweighs group averages, making every mind a unique specimen. On average, the adult male brain is about 10% larger in total volume than the female brain, though this difference does not correlate with general intelligence. The female brain typically possesses a higher percentage of gray matter, which is the tissue responsible for processing information and sensory perception. Men generally have a higher proportion of white matter, the fatty insulation that facilitates long-distance communication between different brain regions. Biology works through subtle shifts in architecture rather than entirely different blueprints. This structural dance begins long before birth, guided by a delicate interplay of genetics and chemical signals. The brain is not a static object. It is a living organ that reshapes itself based on every experience and hormone it encounters. The parietal lobe, involved in spatial perception and navigation, often shows distinct structural variances depending on prenatal hormone exposure. Magnetic resonance imaging reveals that the thickness of the cerebral cortex can vary by region between the sexes, influencing how specific sensory data is filtered. As you listen, the goal is to understand the mechanics of being human without the noise of modern debate. We are looking at the hardware, the physical ridges, the chemical pathways, and the ancient evolutionary pressures that whispered to our ancestors. In many studies, the corpus callosum, which acts as the primary data bridge between the left and right hemispheres, shows a larger posterior section in female subjects. The amygdala, a small almond-shaped structure deep in the temporal lobe, processes emotions and threats with slightly different neural densities in men and women. Viewing the brain through a biological lens allows us to appreciate the diversity of human experience as a natural consequence of evolution. Nature rarely chooses a single path, instead favoring a spectrum of strategies that ensure the survival and adaptability of our species. Survival in the Pleistocene era required a specialized set of cognitive tools that were forged over millions of years of environmental pressure. The ancestral landscape was an unforgiving teacher, rewarding those whose neural pathways were most efficiently tuned to their specific daily survival tasks. While early humans shared the same fundamental goals of safety and sustenance, the practical methods for achieving them often diverged based on the physical demands of their roles. This divergence is often conceptualized through the hunter-gatherer hypothesis, which suggests that distinct cognitive strengths emerged from a natural division of labor. Men who spent weeks tracking mobile prey across vast, unfamiliar territories benefited from a brain optimized for long-distance spatial navigation and three-dimensional mental rotation. This specific pressure likely favored the development of the parietal cortex, the region responsible for calculating the trajectory of a thrown spear or the distance to a fading horizon. Success in hunting required a high degree of systemizing logic, or the ability to understand and predict the physical laws of the external world. Women, who historically managed the complex task of gathering calorie-dense plant foods, faced a different set of neurological challenges. Gathering required an exceptional memory for stationary objects, the ability to distinguish subtle color variations in ripening fruit, and a heightened sensitivity to social cues within the safety of the group. A misidentification of a berry or a failure to read the emotional state of a communal partner could have dire consequences for the survival of offspring. These roles were not rigid boundaries, but they acted as persistent selective pressures that favored certain mental traits over hundreds of thousands of generations. Nature is a master of efficiency and rarely invests energy in traits that do not offer a clear metabolic advantage. The concept of metabolic cost is central here, as the brain is the most energy-expensive organ in the human body, consuming roughly 20% of our total caloric intake. Building and maintaining neural tissue is a massive biological investment that must be justified by its contribution to reproductive success. If a specific cognitive strategy, such as high-velocity motion tracking, was more critical for one segment of the population, evolution would naturally conserve that trait where it was most utilized. Similarly, if verbal fluency and multi-track social monitoring provided a higher survival payoff in a communal setting, those pathways would be prioritized and strengthened. This specialization did not create two different species, but rather two complementary sets of survival strategies that allowed the group as a whole to thrive. The interplay between these different cognitive toolkits ensured that the tribe possessed a wider range of problem-solving abilities than any single individual could hold alone. What began as a response to the harsh realities of the African savanna eventually became encoded in our genetic blueprints. As we move from the ancient history of our ancestors into the modern era, we see these prehistoric echoes in our current biological inheritance. The structural differences we observe in the modern brain are the living fossils of these ancestral pressures, still influencing how we perceive and interact with the world today. Every ridge and fold in the cortex carries the story of a species that learned to survive by diversifying its mental strengths. The human brain manifests its sexual dimorphism through physical characteristics that are often misunderstood or oversimplified in popular culture. Total intracranial volume in men is generally about 10 to 15% larger than in women, a fact that primarily reflects the larger average body size and muscle mass of the male frame. This volumetric difference is purely structural and bears no correlation with general intelligence, as cognitive capacity is determined by the complexity of neural connections rather than raw displacement. A larger container does not necessarily hold a more sophisticated processor, as evidenced by many highly intelligent species with significantly smaller brains than our own. Beneath this difference in scale, the composition of the brain tissue itself reveals a fascinating divergence in how information is prioritized. Male brains typically contain a higher percentage of white matter, which consists of the long, myelinated axons that act as the high-speed cables connecting distant regions of the mind. This abundance of white matter often facilitates efficient communication within a single hemisphere, streamlining the process of translating sensory input into physical action. In contrast, the female brain possesses a higher percentage of gray matter, the densely packed clusters of neuron cell bodies where the actual processing and computation of information occur. This suggests a biological trade-off where one system favors the rapid transit of signals across long distances, while the other prioritizes local processing power and data integration. Symmetry also plays a critical role in distinguishing these two anatomical blueprints, as the male brain tends toward a higher degree of functional asymmetry. In many men, the left hemisphere is more specialized for logical and linear tasks, while the right is more dedicated to spatial awareness, with less crosstalk between the two sides during specific activities. The female brain often shows more bilateral symmetry, utilizing both hemispheres simultaneously for tasks that might be localized to one side in a male counterpart. This structural balance is supported by the density of the neurons themselves, which varies significantly between the sexes. While the male brain may be larger in volume, the female brain frequently displays a much higher packing density of neurons in certain areas of the cerebral cortex. The temporal lobe, which is essential for language and social cognition, often contains more neurons per unit of volume in women than in men. This means that even in a smaller physical space, the female brain can house an equal or sometimes greater number of active processing units. Nature uses this high-density configuration to ensure that the critical functions of communication and emotional intelligence are not compromised by a smaller cranial capacity. It is a masterpiece of biological engineering, proving that efficiency and compact design are just as effective as expansive wiring. The resulting packing density allows the female mind to remain highly interconnected and responsive to a wide array of simultaneous inputs. Understanding these physical foundations helps clarify why the two sexes often approach the same problem using slightly different neurological pathways. The physical bridge that connects the two hemispheres is a thick bundle of nerve fibers known as the corpus callosum. Research from institutions like the University of Pennsylvania has consistently shown that this bridge is often structurally different between the sexes, particularly in its posterior section. In many women, the corpus callosum is significantly more robust, containing a higher density of fibers that allow for rapid communication between the left and right sides of the brain. This increased connectivity facilitates a more integrated approach to processing information, where the logical left and the intuitive right are in constant, high-speed dialogue. Because the two halves of the brain can share data so fluidly, the female mind often finds it easier to synthesize disparate pieces of information into a single, cohesive picture. Extensive neuroimaging studies conducted between 2014 and 2025 have revealed a fundamental divergence in the broader wiring diagrams of the human brain. Male brains typically exhibit a pattern defined by intrahemispheric connectivity, which means the neural pathways run predominantly from the front of the brain to the back within a single hemisphere. This front-to-back wiring creates a direct link between the areas responsible for perception in the rear and those responsible for action in the front. Such a configuration is highly efficient for tasks that require a tight loop between seeing something and doing something, such as tracking a moving object or navigating a complex physical environment. When the brain is wired this way, it operates like a series of specialized, localized networks that can focus intense energy on a single objective without being distracted by peripheral data. Contrast this with the prevailing pattern in female brains, where the connectivity is more interhemispheric or side to side. This lateral wiring weaves the two hemispheres together, allowing for a distributed network that excels at social cognition and multi-track processing. While the male-style wiring is like a high-speed transit line moving in one direction, the female-style wiring is more akin to a vast, interconnected web where every node can talk to every other node simultaneously. This distributed architecture explains why many women are able to monitor multiple streams of information at once, such as listening to a conversation while observing subtle changes in a person's body language. It is a biological strategy that prioritizes the big picture and the context of a situation over the isolated, linear execution of a single task. These wiring differences directly influence how the two sexes handle daily cognitive demands. The front-to-back connectivity in men often leads to a natural aptitude for single-minded focus, where the brain can tune out the world to complete a specific technical challenge. This is sometimes described as a systemizing approach, where the mind seeks to understand the rules and mechanics of a closed system. The side-to-side connectivity in women supports an empathizing approach, where the mind is constantly scanning for social cues and emotional nuances that require the integration of logic and feeling. It is not that one way is superior, but rather that each offers a distinct advantage depending on the environment. Think of the male brain as a collection of highly specialized tools in a workshop, each designed to do one thing exceptionally well. The female brain functions more like a sophisticated command center where every screen is live and the operators are constantly cross-referencing data to maintain a broad awareness of the entire operation. This difference in information flow means that a man might solve a problem by drilling down into the specific mechanics of the issue, while a woman might solve the same problem by looking at how it affects the surrounding social or environmental network. Have you ever noticed yourself leaning toward one of these styles when you are trying to solve a puzzle or manage a busy day? This interplay between localized and distributed networks ensures that as a species, we have both the laser-focused specialists and the broad-minded integrators needed for survival. The architecture of the human mind begins its most dramatic divergence long before a child ever takes their first breath. Around the 8th week of gestation, a tiny chemical signal initiates a process that will permanently alter the physical landscape of the developing brain. In every embryo, the initial blueprint for the brain follows a female-typical pattern, a phenomenon often described in biology as the default female hypothesis. This foundational structure remains largely unchanged unless specific hormonal triggers intervene to redirect its growth toward a different configuration. Between the 12th and 24th weeks of pregnancy, a critical window opens where the fetal testes in males begin to produce a massive surge of testosterone. This event, frequently referred to by neuroscientists as the prenatal testosterone bath, acts as a biological architect, reshaping the neural circuitry that is already under construction. These androgens do not just influence the development of physical traits. They physically prune and expand specific regions of the cerebral cortex. This organizational effect is permanent, meaning the brain is being hardwired with certain predispositions that will persist throughout an individual's entire life. Interestingly, the mechanism for this transformation is remarkably complex, involving a protein called the aromatase enzyme. Once testosterone enters the developing brain cells, this enzyme actually converts much of it into a form of estrogen. It is this localized presence of estrogen within the male brain that paradoxically triggers the masculinization of certain neural pathways, such as the enlargement of the amygdala and the reduction of connectivity in the corpus callosum. Nature provides us with subtle, external clues about the intensity of this internal chemical process through the proportions of the human hand. The ratio between the lengths of the index finger and the ring finger, known as the 2D, 4D digit ratio, is widely studied as a biological marker of prenatal hormone exposure. A longer ring finger relative to the index finger typically correlates with higher levels of testosterone in the womb, which in turn is linked to specific structural variations in the brain's spatial processing centers. This ratio is not a definitive rule for any one individual, but across large populations, it serves as a fascinating window into the second trimester's hidden chemistry. The timing of this hormonal surge is so precise that if it occurs slightly earlier or later, the resulting neural architecture can vary significantly. These variations contribute to the vast spectrum of human personality and cognitive style, ensuring that no two brains are ever truly identical. While the basic, default, structure provides the essential tools for human consciousness, the introduction of these androgens adds a layer of specialized programming. This period of development is less about creating a better brain and more about diversifying the strategies the mind will later use to navigate the world. The second trimester is essentially a silent period of engineering, where the chemical environment of the womb determines how a person will eventually perceive social cues, manage stress, and process spatial information. Every adult carries the structural legacy of those few pivotal months, a permanent record of a chemical conversation that happened in the dark, quiet safety of the beginning. Newborn infants, only 24 hours old, already demonstrate visual preferences that hint at the biological precursors of the adult mind. When presented with a human face and a mechanical mobile of similar size and color, male neonates tend to spend more time tracking the movement of the mobile. Female neonates, by contrast, show a statistically significant preference for gazing at the human face, suggesting an innate orientation towards social stimuli from the very first day of life. This divergence in attention occurs long before any social conditioning or parental influence can take hold, pointing toward a hardwired neurological starting point. Primates provide further evidence for these deep-seated biological predispositions through their own spontaneous toy preferences. In controlled studies of rhesus macaques, researchers found that male monkeys overwhelmingly chose wheeled toys like trucks, while females spent more time interacting with plush dolls. Since these animals have never seen a television commercial or walked through a toy aisle, their choices suggest that certain shapes and functions appeal to the brain's underlying architecture. The male brain's attraction to mechanical objects may be linked to a predisposition for systemizing and understanding three-dimensional motion. The female brain's affinity for the doll-like form likely reflects an early sensitivity to social agents and the nurturing behaviors essential for group cohesion. Do you remember which toys you gravitated toward most as a young child, and whether they felt like a natural choice or something encouraged by those around you? During the toddler years, the rate of neural development begins to follow distinct timelines depending on the child's sex. Female brains typically prioritize the maturation of the left hemisphere, specifically the areas dedicated to language and fine motor skills. This often results in girls speaking their first words earlier and developing a more extensive vocabulary by the age of 2 compared to their male peers. The density of neurons in the temporal lobe, which processes auditory information and speech, is frequently higher in young girls during this window. This early verbal advantage allows for more complex social negotiation and a greater reliance on communication to resolve conflicts. Male toddlers often exhibit a different developmental trajectory, with a greater emphasis on gross motor skills and spatial awareness. This physical drive manifests as rough and tumble play, a behavior observed across nearly all mammalian species. Far from being random aggression, this type of play serves as a vital neurological rehearsal for navigating physical hierarchies and testing personal limits. It strengthens the cerebellum and the motor cortex, refining the brain's ability to coordinate large-scale movements and judge distances. This period of childhood acts as a foundation, a time when the brain's structural blueprints are tested and reinforced through daily experience. As these early childhood patterns stabilize, the brain prepares for the most dramatic restructuring it will ever undergo. The relatively steady growth of the early years is about to be interrupted by a massive biological upheaval. This transition marks the end of childhood's quiet preparation and the beginning of a profound neural transformation. The brain is moving toward a crucible of change that will redefine its chemistry and its very sense of self. This upcoming shift will sharpen the differences between individuals, pushing the mind into the intense, high-stakes environment of adolescence. The brain undergoes a massive architectural renovation during the onset of puberty, a process defined by the aggressive streamlining of neural connections. Synaptic pruning acts as a biological sculptor, removing weaker or less used synapses to make the remaining pathways faster and more efficient. While this happens in all adolescents, the timing and location of this pruning vary significantly between the sexes. In female brains, this refinement often begins and concludes earlier, particularly in the regions governing social cognition and impulse control. Male brains typically experience a more prolonged period of pruning, which extends well into the early 20s, specifically within the areas dedicated to spatial reasoning and sensory integration. This divergence creates a temporary but profound gap in how the two sexes process the world during these formative years. A second wave of hormonal organization begins to wash over the brain, cementing the structural blueprints established in the womb. This surge of estrogen or testosterone acts like a chemical sealant, hardening certain neural pathways while allowing others to remain plastic. The prefrontal cortex, often described as the brain's CEO, handles complex decision-making and the modulation of intense emotions. Research indicates that this region generally reaches full maturity several years earlier in females than in males. This discrepancy helps explain why adolescent girls often exhibit a more advanced capacity for long-term planning and social navigation during their mid-teens. The male prefrontal cortex remains in a state of flux longer, leaving the emotional centers of the brain with less oversight during a critical window of development. The introduction of the menstrual cycle brings a rhythmic, cyclical nature to the architecture of the female brain. Studies using magnetic resonance imaging have shown that hippocampal volume actually fluctuates in response to changing estrogen levels throughout the month. The hippocampus, which is vital for memory and emotional regulation, can expand or contract slightly depending on the phase of the cycle. This high level of plasticity suggests that the female brain is constantly recalibrating its emotional and cognitive resources in a way the male brain is not. These subtle shifts in brain volume are not signs of instability, but rather evidence of a highly responsive and adaptive neurological system. Adolescent males face a different biological challenge as their testosterone levels spike to nearly 20 times their childhood baseline. This massive hormonal influx has a direct and measurable impact on the amygdala and the ventral striatum, the brain's primary reward and threat centers. Testosterone decreases the sensitivity of the brain's risk assessment hardware, making the potential for a reward seem far more significant than the possibility of a negative consequence. This neural environment encourages the exploration of boundaries and the pursuit of status, which were historically necessary for survival in tribal hierarchies. The result is a brain that is temporarily optimized for action and competition, even when the prefrontal brakes are not yet fully installed. This period of divergence creates a landscape where two different sets of survival strategies are being refined simultaneously. The brain is no longer a generalist. It is becoming a specialist, tuned by its own internal chemistry to navigate specific social and physical environments. These changes are not merely psychological but are etched into the very density of the white matter and the thickness of the cortex. As the turbulent years of puberty begin to level off, the mind emerges with a distinct set of tools for processing reality. This transformation sets the stage for the specific chemical drivers that will dominate adult life. One of these drivers is a powerful molecule that influences everything from physical strength to the way a person perceives a challenge. Testosterone operates within the adult male brain much like a primary neurotransmitter, traveling through the bloodstream to dock with specialized receptors in the amygdala and the hypothalamus. This molecular interaction subtly shifts the brain's baseline, prioritizing the pursuit of status and social standing over immediate comfort. High concentrations of these receptors in the reward centers mean that achieving a goal or gaining respect triggers a more intense dopamine release. Scientific observations suggest that this chemical drive is less about raw power and more about the preservation of one's position within a hierarchy. When a man feels his status is being acknowledged, the brain maintains a steady, regulated state of focus. The challenge hypothesis, a well-regarded concept in behavioral biology, explains that testosterone levels are not static but highly reactive to the environment. Levels often surge during a competitive event and remain elevated in the aftermath of a victory, providing a neurological winner effect that encourages further persistence. Conversely, a significant social or physical loss can cause these levels to dip, prompting the brain to adopt a more cautious, low-profile strategy to avoid further conflict. This internal feedback loop ensures that the individual's behavior remains calibrated to their current social reality. It is a sophisticated system of biological accounting that balances the energy cost of competition against the potential for reward. A common misunderstanding in popular culture is the conflation of testosterone with simple aggression, yet the reality is more nuanced. Dominance, in a biological sense, refers to the drive to achieve a specific outcome or maintain a leadership role, which often requires high levels of cooperation and social intelligence. The hormone facilitates a state of focused goal pursuit, allowing the brain to tune out distractions and remain committed to a long-term objective. In many social species, the individuals with the highest status are often the most composed, using their influence to stabilize the group rather than disrupt it. This goal-oriented persistence is a hallmark of the androgen-influenced mind. Contrary to the myth of testosterone rage, the hormone often has a profound calming effect on the male nervous system when levels are within a healthy, natural range. It can act as a buffer against anxiety by modulating the way the brain processes fear signals in the amygdala. When testosterone is low, men often report feeling more irritable, anxious, and less capable of handling daily stressors. This suggests that the molecule provides a sense of emotional steadiness and confidence that is essential for decisive action. By reducing the background noise of social apprehension, the brain is free to dedicate its energy to external problem solving and navigation. This chemical environment creates a cognitive profile that favors decisive, single-track thinking over broad, multi-layered processing. The male brain, bathed in this steady influence, becomes a tool for specialized application, whether that involves physical labor, complex system analysis, or strategic planning. The drive for status ensures that these tasks are not just performed, but performed with a high degree of competence and pride. While this focus is powerful, it is only one part of the human story, as other molecules work in parallel to protect and maintain the brain's structural integrity. These protective elements ensure that the high energy demands of a focused life do not lead to premature neurological wear. Estrogen serves as a powerful natural shield for the brain, functioning as a neuroprotectant that promotes the survival and health of neurons. This hormone actively encourages synaptic plasticity, which is the brain's ability to form new connections and reorganize itself in response to learning or environmental changes. Beyond its role in reproduction, estrogen influences the production of brain-derived neurotrophic factor, a protein often described as fertilizer for the mind. This biological support system helps maintain the density of dendritic spines, the tiny protrusions on neurons that receive signals from other cells. Without this consistent hormonal support, the communication between different brain regions would likely become less efficient over time. Verbal memory and fine motor skills receive a significant boost from the presence of estrogen in the female brain. Research in the journal Nature Neuroscience suggests that higher levels of this hormone correlate with an increased ability to recall complex word lists and execute delicate manual tasks. This explains why certain cognitive strengths often peak at specific points in the hormonal cycle when estrogen is most abundant. The hormone stimulates the hippocampus, a region critical for memory, ensuring that information is encoded and retrieved with a high degree of precision. These subtle shifts in ability demonstrate how deeply biological rhythms are integrated into everyday mental performance. Progesterone introduces a different kind of influence, acting as a natural sedative that stabilizes the brain's electrical activity. This molecule interacts with GABA receptors, the primary inhibitory neurotransmitters responsible for reducing excitability and inducing a sense of calm. When progesterone levels rise, the brain often enters a state of increased emotional regulation and improved sleep quality. This monthly architecture means the female brain undergoes a structural and chemical renovation every few weeks, rather than remaining in a static state. These fluctuations are not disruptions, but rather a sophisticated balancing act between high-energy cognitive drive and restorative neurological rest. The ovulation cycle creates a series of predictable cognitive shifts that alter how a person interacts with the world. During the follicular phase, rising estrogen often leads to heightened social outgoingness, faster verbal processing, and an increased sensitivity to environmental rewards. Once ovulation passes and progesterone takes the lead, the focus often shifts toward internal monitoring and a more cautious, detail-oriented approach to problem-solving. Some studies indicate that even spatial navigation strategies can change throughout the month, moving between landmark-based and direction-based methods depending on the hormonal mix. This dynamic flexibility allows the brain to excel at different types of challenges at different times. Do you find that your own mental energy or focus tends to shift in a rhythmic pattern throughout the month? While the male brain operates on a relatively stable hormonal baseline with a slow, linear decline over decades, the female brain thrives on a cyclical baseline. This constant ebb and flow of estrogen and progesterone creates a unique neurological environment that is always adapting and recalibrating. Men typically experience a 24-hour testosterone cycle, whereas women navigate a much longer and more complex chemical journey. This difference in timing means that while one system favors a steady, unchanging output, the other is optimized for periodic peaks in specific cognitive domains. Understanding these protectors reveals that the mind is never truly the same from one week to the next. Deep within the hypothalamus, a different chemical narrative begins to take shape through the release of two powerful neuropeptides that define how we connect with others. Oxytocin is frequently described as the cuddle chemical, yet its influence on the brain is far more nuanced and structurally dependent on biological sex. In the female brain, a higher density of oxytocin receptors in regions like the amygdala and the ventromedial prefrontal cortex creates a profound sensitivity to social touch and emotional warmth. This high receptor availability means that even small amounts of the hormone can trigger an intense feeling of safety and trust. For men, the story of social bonding often relies more heavily on a closely related molecule called vasopressin. While oxytocin promotes a sense of open, communal connection, vasopressin is fundamentally linked to a more vigilant form of attachment. In many male mammals, including humans, vasopressin levels rise during the formation of a pair bond, but its primary role is to activate the brain's circuitry for protection and territoriality. This chemical drives a desire to guard the home and the partner, shifting the brain's focus toward identifying external threats rather than purely internal warmth. It is this subtle chemical distinction that helps explain why the male experience of love often manifests as a protective drive. Scientists have observed that while oxytocin softens the edges of the ego to allow for intimacy, vasopressin sharpens the focus on the boundaries of the family unit. These hormonal leanings create two distinct survival strategies known as the tend and befriend model and the defend and protect model. When faced with a crisis, the female brain often utilizes oxytocin to lower the heart rate and seek out social support, a strategy that historically increased the survival of offspring. This biological impulse to gather the group and share resources acts as a natural buffer against the damaging effects of cortisol. Conversely, the male brain's reliance on vasopressin often leads to a physical and mental hardening in the face of stress. Instead of seeking a circle of support, the vasopressin-led response prioritizes securing the perimeter and preparing for a direct confrontation. Does your first instinct during a stressful moment involve reaching out to a friend, or do you prefer to handle the situation in quiet isolation? The interaction between these chemicals and our primary sex hormones further separates these two paths. Estrogen tends to enhance the effects of oxytocin, making the female brain more resilient to isolation through the seeking of social bonds. Testosterone, however, can sometimes dampen the effects of oxytocin while amplifying the competitive aspects of vasopressin. This does not mean that men do not feel deep affection, but rather that their biological architecture processes trust through the lens of shared loyalty and mutual defense. In the female brain, trust is often built through the exchange of emotional vulnerability and verbal transparency. In the intricate mechanics of long-term pair bonding, these molecules work together to ensure the continuity of the species. The female brain's oxytocin-heavy system ensures a deep, intuitive connection to the needs of others, while the male brain's vasopressin system provides the structural stability and vigilance required for external security. Even the way trust is repaired after a conflict differs, as one system seeks a return to emotional harmony and the other seeks a return to functional order. This chemical partnership ensures that whether the environment requires a soft hand or a watchful eye, the human brain is equipped to provide it. Understanding these neuropeptides reveals that bonding is not a single, universal feeling, but a specialized biological toolkit. Deep within the temporal lobes of every human brain sits a pair of almond-shaped structures known as the amygdala. This ancient cluster of nuclei serves as the brain's primary alarm system, scanning the environment for threats and assigning emotional significance to every experience. Research in neuroanatomy consistently reveals that the amygdala is typically larger in the male brain, even when accounting for overall differences in total brain volume. This size disparity translates to a heightened sensitivity toward physical cues and external challenges, often resulting in a more immediate physiological response to perceived danger. In the male brain, The right amygdala tends to be more active, a region specifically linked to the encoding of action-oriented emotional memories. When a man experiences a high-stress event, his brain prioritizes the what and how of the situation, focusing on the physical movements and external variables required for a quick resolution. The female brain displays a different pattern of activation, often showing a more robust response in the left amygdala. This side of the alarm system is closely associated with detail-oriented emotional memory and the processing of internal states. While the male brain might remember the trajectory of a threat, the female brain is biologically tuned to remember the specific facial expressions, tone of voice, and nuanced context surrounding an emotional event. These structural differences create a distinct contrast in how the two sexes process and store the history of their lives. For the male, an emotional memory is a blueprint for future action, while for the female, it is a rich, multi-layered narrative of interpersonal connection and subtle social cues. This divergence in neural architecture also influences the timing and duration of a stress response, often described as the short fuse versus the long burn. Because the male amygdala is so tightly coupled with the motor centers of the brain, the reaction to a threat is often explosive and brief. This is a survival mechanism designed for immediate physical defense, where a surge of energy is followed by a rapid return to a physiological baseline once the threat is neutralized. In contrast, the female amygdala maintains a more sustained level of activation, leading to a response that may develop more slowly but persists for a longer duration. This long burn allows the female brain to remain vigilant over a longer period, ensuring that every detail of a potential problem is thoroughly analyzed and integrated into the social memory of the group. The relationship between the amygdala and the prefrontal cortex further distinguishes these two biological strategies. The prefrontal cortex acts as the brakes of the brain, providing the executive control necessary to dampen the amygdala's raw emotional impulses. In the female brain, the connection between the prefrontal cortex and the amygdala is often more direct and structurally dense. This enhanced, top-down control allows for a more fluid integration of emotion and reason, facilitating the ability to talk through a feeling rather than acting on it physically. The male brain typically possesses a more detached relationship between these two regions, which can lead to a momentary bypass of the analytical centers during times of extreme provocation. Science suggests that these different wiring patterns are not about one being superior to the other, but about diversifying the ways a human group can survive a crisis. The male brain's larger right amygdala provides the decisive, fast-acting energy needed for protection and competition. Simultaneously, the female brain's left hemisphere focus provides the careful, detail-rich emotional intelligence required to navigate complex social landscapes and avoid conflict before it begins. These internal alarm systems work in tandem across the species, ensuring that no threat goes unnoticed and no lesson is forgotten. The interplay between the impulsive fire of the amygdala and the cooling influence of the cortex remains one of the most fascinating chapters in the study of our shared biology. Deep within the temporal lobe lies the hippocampus, a curved structure named for its resemblance to a seahorse that serves as the brain's primary engine for memory and spatial awareness. While the amygdala handles the raw fire of emotion, the hippocampus acts as the librarian, carefully indexing experiences into long-term storage and creating the internal maps we use to navigate the physical world. Research in neuroscience, including studies cited in the journal Nature, indicates that although the male brain is typically larger in total volume, the female hippocampus is often larger relative to the overall size of the brain. This structural prominence suggests a heightened biological investment in the specific types of processing this region governs, particularly in the realm of social and contextual memory. The way we move through a landscape reveals a profound divergence in how these neural maps are constructed and utilized by the different sexes. Men frequently utilize an egocentric navigation strategy, which relies on a mental sense of direction, dead reckoning, and an internal compass of Euclidean distance. This method allows a person to orient themselves toward a goal even when the surrounding environment is unfamiliar or lacks clear markers. In contrast, the female brain typically favors an allocentric navigation strategy, which is heavily dependent on landmarks and the relative positions of objects within a space. This approach relies on recognizing a specific tree, a distinct building, or a bend in a path to determine one's location, creating a rich, detail-oriented tapestry of the environment. This reliance on landmarks is closely linked to a cognitive skill where women consistently demonstrate a significant biological advantage: object location memory. From an evolutionary perspective, the ability to remember exactly where a specific calorie-dense plant was growing or where a tool was left would have been a vital survival trait for a gatherer. The female hippocampus appears specifically tuned to notice and retain the precise placement of items within a complex visual field. While a male brain might excel at calculating the shortest geometric route across a mountain range, the female brain is often superior at recalling the specific arrangement of objects within a room or the exact spot where a key was placed days prior. Memory is rarely a neutral recording of facts. It is almost always colored by the chemical state of the brain at the moment an event occurs. Because the hippocampus is so tightly integrated with the emotional centers of the limbic system, it plays a central role in how we remember the feeling of a situation. The female brain typically encodes emotional memories with greater vividness and more granular detail than the male brain. This often results in a more robust autobiographical memory, where the specifics of a conversation, a social slight, or a moment of connection are preserved with remarkable clarity over many years. Does your own memory tend to favor the big picture of an event, or do you find yourself focusing on the specific words and sensory details? This enhanced encoding is thought to be influenced by the way estrogen interacts with hippocampal neurons, promoting a high degree of synaptic plasticity. By weaving emotion and fact together into a single, durable thread, the hippocampus ensures that social lessons are learned deeply and recalled instantly when similar situations arise. This creates a biological foundation for a sophisticated social intelligence that relies as much on the past as it does on the present. Through this intricate interplay of navigation and remembrance, the brain ensures we never truly lose our way, whether in the physical world or the complex landscape of human relationships. Spatial navigation is not a singular skill, but a collection of distinct mental operations that rely on different regions of the parietal and occipital lobes. One of the most consistently observed differences in human cognition involves the mental rotation of three-dimensional objects. When tasked with visualizing a complex shape and predicting how it would look if turned 45 degrees in space, the male brain generally exhibits a faster response time and higher accuracy. This capability is deeply rooted in the structural layout of the parietal cortex, which manages the spatial relationship between the self and the external environment. The evolutionary pressures of hunting required an acute ability to calculate trajectories and intercept moving targets across vast, changing landscapes. Tracking the arc of a projectile or predicting the path of a fleeing animal necessitated a neural architecture capable of rapid geometric modeling. This specific environmental demand likely shaped the male parietal lobe to prioritize egocentric navigation, which focuses on the distance and direction of the body relative to a target. Because these tasks were matters of life and death over hundreds of thousands of years, the male brain developed a specialized efficiency for rotating objects in the mind's eye to foresee their movement through physical space. This spatial prowess extends into the realm of systemizing, a cognitive style that seeks to understand the underlying rules of mechanical and physical systems. The male brain often demonstrates a natural inclination toward analyzing how parts of a machine interact or how a complex structure maintains its balance. This is not merely a socialized preference, but a reflection of a brain organized to prioritize the if-then logic of the physical world. By focusing on the mechanics of things rather than the nuances of social interaction, this systemizing drive allows for the creation and manipulation of tools with a high degree of precision. Targeting and interception represent the pinnacle of this spatial specialization, involving a near-instantaneous synchronization between the eyes and the motor cortex. Men frequently show a biological advantage in tasks requiring the tracking of multiple moving objects at once, such as during a hunt or a modern-day high-speed navigation task. This speed of processing in the visual spatial pathways allows for the rapid adjustment of physical movements in response to a changing environment. The ability to aim effectively is a complex neurological feat that involves estimating wind, gravity, and velocity, all within a fraction of a second. While the male brain excels at calculating distance and rotation, the female brain demonstrates a distinct superiority in the memory of spatial locations and the identification of objects. In the context of a gathering-based economy, it was far more valuable to remember the exact location of a specific plant or a hidden water source within a dense forest. This object location memory allows the female brain to maintain a highly detailed map of the environment that includes specific landmarks and their various properties. Rather than focusing on the geometry of the route, this strategy relies on a rich, descriptive understanding of the landscape that ensures nothing of value is lost or forgotten. Color memory and discrimination also play a critical role in this female spatial strategy, as the ability to distinguish between subtle shades of red and green could mean the difference between finding ripe fruit and consuming something toxic. The female visual system is often more sensitive to the nuances of the color spectrum, particularly in the longer wavelengths. This heightened color perception, combined with a superior memory for texture and detail, creates a unique form of spatial awareness that is descriptive rather than purely geometric. By balancing the male focus on trajectory and rotation with the female focus on location and identification, the human species developed a comprehensive toolkit for navigating the world. The human capacity for complex speech relies on a sophisticated coordination of muscle control and rapid cognitive retrieval. Female brains typically exhibit a higher degree of verbal fluency, which manifests as the ability to generate a larger number of words within a specific category in a short window of time. This biological advantage is not merely a matter of practice, but is supported by the physical density of the cortex in areas dedicated to communication. Neurological imaging reveals a striking difference in how the two sexes process the spoken word. When engaged in linguistic tasks, the female brain often displays bilateral activation, meaning both the left and right hemispheres work in tandem to decode meaning and tone. In contrast, the male brain tends toward lateralization, utilizing the left hemisphere almost exclusively for the structural elements of language. This dual hemisphere approach in women allows for a more integrated processing of both the literal definition of words and the emotional context behind them. How often have you noticed the subtle shift in someone's tone before they even finish their sentence? Deep within the temporal and frontal lobes lie the primary hubs for human communication, known as Wernicke's and Broca's areas. Research in the Journal of Neuroscience has documented that these regions contain significantly higher neuron density in female brains compared to males. Specifically, the layers of the cortex that handle the reception and processing of speech are more tightly packed with cells, facilitating faster signal transmission. This structural reinforcement ensures that the female brain can manage multiple streams of verbal information with high precision. Social survival historically depended on reading unspoken intentions. The female brain demonstrates a heightened sensitivity to nonverbal cues, driven by increased activity in the mirror neuron system and the superior temporal sulcus. This allows for the near-instantaneous detection of subtle facial microexpressions that might pass unnoticed by a more task-oriented male observer. These biological sensors transform a simple conversation into a multi-layered exchange of emotional data and social feedback. At the molecular level, speech is guided by genetic markers. The FOXP2 gene, frequently referred to as the language gene, plays a vital role in the fine motor movements required for vocalization. A study by the University of Maryland School of Medicine found that the FOXP2 protein is significantly more abundant in the brains of young girls than in boys. This chemical disparity likely explains why female children often reach language milestones earlier and with greater complexity during early development. The architectural layout of the language centers directly influences social cohesion. While the male brain may focus on the transmission of factual data, the female brain is biologically tuned to maintain the social fabric through nuanced dialogue. This divergence in communication style is a reflection of the different evolutionary pressures placed upon the sexes throughout human history. By understanding these biological roots, we can better appreciate the diverse ways in which humans connect with one another. The human retina functions as a sophisticated filter, sorting light into different streams of data before it ever reaches the visual cortex. Within this biological layer, two primary types of ganglion cells, known as M cells and P cells, dictate how we perceive the world. Male eyes are characterized by a greater density of M cells, or magnocellular cells, which are physically larger and more efficient at detecting rapid movement. These cells do not prioritize color or fine detail, but instead focus on the speed and trajectory of an object. Such specialization provides a biological advantage in tracking motion and calculating depth in three-dimensional space. Contrastingly, the female retina contains a significantly higher concentration of P cells, or parvocellular cells, which are smaller and more numerous. These cells are designed to capture high-resolution details, intricate textures, and a vast array of colors. While the male brain might track the flight of an object through the air, the female brain is biologically optimized to identify the specific characteristics of that object once it is stationary. Focusing on the what, rather than the where, allows for a much more detailed internal map of the immediate environment. Color perception is not distributed equally across the species, as the genes for red and green photopigments are located on the X chromosome. Because women possess two X chromosomes, they often have a more diverse and sensitive array of color receptors. Such a genetic configuration leads to a heightened sensitivity to the red-orange spectrum, allowing the female brain to distinguish between subtle shades that might appear identical to a man. Historically, this capability was essential for identifying ripe fruit or distinguishing edible berries from poisonous ones against a backdrop of green foliage. Do you find yourself noticing small changes in the arrangement of a room that others seem to miss entirely? Such observational skill is often linked to the breadth of the visual field and how it is processed. The female visual system typically offers a wider peripheral awareness, acting like a wide-angle lens that monitors the entire environment for subtle shifts. In contrast, the male visual system tends to favor a more narrow, foveal focus, often referred to as tunnel vision. A concentrated gaze allows for intense focus on a single point of interest, filtering out peripheral distractions to ensure the primary target remains clear. These differences in visual processing reflect the specialized roles our ancestors occupied for thousands of generations. The male brain's reliance on motion tracking supported the high-stakes coordination required for hunting and long-distance navigation. Meanwhile, the female brain's emphasis on color and texture supported the complex task of gathering, foraging, and social monitoring. These two distinct ways of seeing do not compete, but rather combine to give the human species a comprehensive and highly adaptable understanding of its surroundings. Beyond the visual world, the human nervous system interprets the physical environment through a sophisticated network of receptors that register touch and discomfort. Research in neurobiology has revealed that the pathways used to process pain signals are not identical between the sexes, leading to different physiological experiences of the same stimulus. In the female body, there is a significantly higher density of nerve fibers within the skin, particularly in the face and extremities. This structural difference results in an increased sensitivity to tactile stimuli, allowing the female brain to detect subtle textures or pressures that a male brain might overlook. Estrogen plays a central role in how these signals are modulated before they ever reach the conscious mind. This hormone acts as a natural regulator, often lowering the threshold for pain during specific phases of the hormonal cycle when levels are in flux. When estrogen is high, it can enhance the brain's sensitivity to certain types of discomfort, while its decline can sometimes trigger inflammatory responses that the nervous system perceives more acutely. Because of this cyclical influence, the experience of physical sensation for women is often more dynamic and variable than the relatively steady baseline found in men. Men typically exhibit a different set of neurological adaptations for managing intense physical stress, a phenomenon known as stress-induced analgesia. During moments of high-stakes competition or physical confrontation, the male brain can more readily suppress pain signals through a rapid release of endogenous opioids and adrenaline. This mechanism allows for continued action and focus even when the body has sustained an injury, prioritizing immediate survival over the recognition of damage. While this provides a short-term evolutionary advantage in dangerous environments, it can also lead to a delay in seeking necessary medical attention for underlying issues. Recent findings published between 2024 and 2025 have shed new light on the memory of pain, suggesting that the sexes encode chronic discomfort through different cellular mechanisms. In these studies, researchers discovered that male mice and humans appear to remember painful experiences through the activation of microglia, which are specialized immune cells in the brain and spinal cord. Conversely, the female nervous system seems to bypass these specific cells, utilizing T cells to process the same chronic signals. This divergence suggests that the very foundation of how we suffer and recover is rooted in distinct biological architectures. Have you ever noticed how your own tolerance for discomfort seems to change depending on your mood or the time of day? This subjective experience is tied to how the brain integrates sensory data with emotional context. In the female brain, there is often a stronger connection between the centers that process physical pain and those that manage emotional regulation, such as the anterior cingulate cortex. This integration means that physical and emotional distress are more likely to be processed as a unified experience. For the male brain, these systems are often more compartmentalized, leading to a tendency to treat physical pain as a purely mechanical problem to be solved rather than a holistic state of being. The human experience of sound reveals a distinct landscape where biological architecture shapes how we hear the world. Research consistently shows that the female brain possesses a significant advantage in auditory processing, particularly when it comes to the detection of higher frequencies. This sensitivity is not merely a matter of ear anatomy, but involves the way the primary auditory cortex decodes sound waves into meaningful information. Women generally exhibit a lower threshold for hearing, meaning they can detect faint sounds that often go entirely unnoticed by the average male. This acute hearing extends into the nuances of human speech, where the female brain excels at distinguishing subtle shifts in voice tones and emotional prosody. While a male brain might focus primarily on the literal content of a sentence, the female brain is often simultaneously mapping the speaker's emotional state through pitch, rhythm, and inflection. This ability to read between the lines of a conversation is supported by a higher density of neurons in the language and hearing centers of the temporal lobe. A similarly profound divergence exists within the sense of smell, where the differences are physically visible under a microscope. Studies involving post-mortem analysis have revealed that women possess a significantly higher number of cells in the olfactory bulb, the first brain structure to process odors. In some instances, the female brain contains up to 50% more neurons in this region than the male brain. This biological investment suggests that for much of human history, a heightened sense of smell was a critical survival tool. From an evolutionary perspective, this sensitivity likely served a dual purpose, protecting the family by detecting the subtle scent of spoilage in food and identifying kin through pheromonal signatures. For a nursing mother, the ability to recognize her infant's unique scent is a powerful neurological anchor that facilitates bonding and protection. Because the female brain is often more attuned to these environmental inputs, it can become more easily overwhelmed by sensory noise. A crowded room with overlapping conversations, bright lights, and competing scents can trigger a more rapid stress response in women than in men. This sensory input overload is processed through the limbic system, where the brain must decide if a stimulus is a threat or merely background data. When the environment becomes too loud or too pungent, the amygdala may signal a state of hyperarousal, leading to a feeling of exhaustion or irritability. The male brain, by contrast, tends to filter out these ambient details more aggressively, focusing on a single stream of information while ignoring the peripheral chaos. This filtering mechanism allows for intense concentration in noisy environments, though it may result in missing the subtle social or environmental cues that the female brain captures effortlessly. When the brain perceives a threat, it initiates a chemical cascade that fundamentally alters how a person interacts with the world. This process begins in the hypothalamus, which signals the adrenal glands to release cortisol, the body's primary stress hormone. While both sexes rely on cortisol for survival, the way this chemical interacts with other hormones creates two distinct psychological landscapes. In the male brain, a surge of cortisol often pairs with high levels of testosterone, which can amplify the classic fight-or-flight response. This reaction is deeply rooted in human history, designed to prepare the body for immediate, high-stakes physical action. Research conducted at the University of California, Los Angeles, suggests that the male stress response is more likely to activate the sympathetic nervous system, leading to a state of hyper-focused aggression or a rapid retreat. This physiological state causes the prefrontal cortex to temporarily take a back seat while the amygdala takes control of the steering wheel. During these moments, the male brain often engages in compartmentalization, a survival strategy that pushes secondary emotions and complex social considerations aside to focus entirely on the immediate problem. This singular focus is highly effective for solving mechanical or physical crises, as it prevents the mind from being paralyzed by the gravity of the situation. A different biological script often unfolds within the female brain, where the stress response is frequently moderated by oxytocin. This shift in chemistry has led researchers to identify a secondary survival strategy known as the tend and befriend response. Instead of preparing for a physical confrontation, The female brain may prioritize social connection and the protection of offspring as a means of ensuring safety. The release of oxytocin during times of duress can actually encourage a seeking of consensus and community, using social cohesion as a shield against the perceived threat. This tendency highlights the brain's ability to use cooperation as a tool for resilience, turning a moment of fear into an opportunity for strengthening group bonds. The structural connectivity of the brain also dictates how these two groups process the aftermath of a stressful event. Because the female brain generally possesses more interhemispheric connections, it is prone to a phenomenon known as rumination. This involves the mind repeatedly circling back to the stressor, analyzing every detail and verbalizing the experience to gain a sense of control. While this helps in long-term emotional processing, it can keep the stress response active for a longer period. Men, possessing more front-to-back wiring within each hemisphere, often prefer to move into a task-oriented state to burn off the residual energy of the stress. Does your mind tend to find peace by talking through a difficult day with a friend, or do you prefer to quietly lose yourself in a solitary hobby? This difference in processing is also reflected in the clearance rate of stress hormones from the bloodstream. Scientific observations indicate that once a threat has passed, the male physiology often returns to its baseline state more rapidly than the female physiology. The cortisol levels in men tend to drop sharply once the battle is perceived to be over, allowing for a quicker period of physical recovery. In contrast, the female brain may maintain elevated levels of stress chemicals for a longer duration, as the mind continues to scan the social and environmental horizon for lingering ripples of the conflict. This sustained state of arousal in the female brain is not a flaw, but rather a sophisticated monitoring system designed to prevent the recurrence of the problem. It allows for a deep, reflective analysis of what went wrong and how to avoid it in the future, even if it comes at the cost of temporary exhaustion. The male brain's ability to shut off the stress response quickly is equally vital, as it preserves metabolic energy and allows the individual to return to a state of readiness for the next challenge. These two strategies represent a biological division of labor that has allowed human communities to survive both acute physical dangers and the long-term complexities of social living. Biological research indicates that when processing distress, the female brain shows a higher tendency toward internalizing, which often manifests as reflecting inward on one's own emotional state. This internal focus is linked to higher activity in the subgenual anterior cingulate cortex, a region involved in emotional regulation and self-reflection. Conversely, the male brain is statistically more likely to externalize emotional responses, often directing the energy of a feeling toward physical action or environment-based adjustments. These divergent paths are not indicative of the strength of the emotion, but rather the structural route the signal takes through the brain's architecture. Studies in neurophysiology have measured the speed at which signals travel from the limbic system, the ancient emotional center, to the prefrontal cortex, where rational thought occurs. In many male brains, there is a measurable lag between the initial emotional surge and the cortical processing that follows, which can occasionally lead to a brief period of unnamed feeling. Female brains often exhibit a more rapid integration between these two areas, allowing for a quicker verbal identification of the specific emotion being felt as the signal reaches the language centers almost instantly. The phenomenon known as instrumental grieving describes a cognitive strategy where an individual focuses on solving the practical problems associated with a loss or stressor rather than the feeling itself. Male brains frequently default to this mode, utilizing the parietal lobes to analyze the situation for actionable steps. This is why a man might respond to a friend's distress by offering a tangible solution or a plan of action, as his neural pathways are wired to prioritize environmental mastery over emotional venting. For the female brain, the act of vocalizing an emotion serves a biological purpose beyond simple communication. It is a form of cognitive processing that utilizes the language centers to organize chaotic feelings into a linear narrative. Seeking social validation allows the female brain to lower amygdala activation through the release of calming neuropeptides like oxytocin. This shared emotional experience acts as a regulatory tool, helping the individual transition from a state of high arousal to a state of calm by distributing the cognitive load of the experience. The long-standing cultural myth that one sex is more emotional than the other is not supported by physiological data regarding the actual intensity of internal feelings. Research using functional MRI scans shows that both sexes experience high-intensity activation in the limbic system when exposed to evocative stimuli. The difference lies almost entirely in the downstream expression and the neural pathways used to manage those feelings, rather than the raw capacity for emotion itself. A quiet response in the male brain does not signal a lack of feeling, just as a verbal response in the female brain does not signal a lack of control. This sophisticated machinery of the mind ensures that within any human group, there are individuals who can offer deep empathy and others who can offer steady, objective action. By utilizing different circuits to process the same world, the species maintains a balanced response to the myriad challenges of life. The calm complexity of these interactions serves as a reminder that our biological differences are less about conflict and more about the diverse ways we navigate our shared human experience. Beneath the surface of our social interactions lies a sophisticated dual-track system known as empathy, which functions through two distinct neurological pathways. Cognitive empathy represents the brain's ability to intellectually understand another person's perspective or mental state, essentially allowing an individual to map out what someone else is thinking. Affective empathy, conversely, is the visceral capacity to actually feel the emotions of another, mirroring their distress or joy within one's own nervous system. While every healthy human brain utilizes both, the balance between these two systems often leans in different directions based on biological sex. This divergence is central to the systemizing empathizing theory, which suggests that brains generally fall along a spectrum between a drive to analyze systems and a drive to identify with emotions. Systemizing involves a deep focus on the underlying rules that govern objects, machines, or abstract ideas to predict how they will behave. Empathizing focuses on the spontaneous, often unpredictable cues of human behavior to foster social connection and mutual understanding. Research from the University of Cambridge indicates that while individuals of any gender can be high systemizers or high empathizers, the male brain is statistically more likely to prioritize systemizing, while the female brain shows a stronger predisposition toward empathizing. The physical hardware for these experiences resides in the mirror neuron system, a network of cells that fire both when we perform an action and when we observe someone else performing that same action. In the female brain, these mirror neurons tend to be more highly sensitive and integrated with the limbic system, leading to a more immediate and frequent resonance with the feelings of others. This creates a baseline of continuous social monitoring, where the female brain is often scanning the environment for subtle shifts in tone, posture, or facial expressions. This constant data stream allows for a high degree of social cohesion and the ability to anticipate the needs of a group before they are even voiced. In contrast, the male brain demonstrates a unique neurological trait characterized by the ability to temporarily suppress or gate the empathy centers when a task requires analytical focus. When a man is faced with a problem that requires objective, mechanical, or tactical thinking, his brain can reduce the activity in the areas responsible for effective empathy. This is not a lack of caring, but rather a functional shift that allows the prefrontal cortex to operate without the interference of emotional static. This gating mechanism is particularly useful in high-stress environments where making a detached, logical decision is necessary for survival or the completion of a complex objective. Have you ever noticed how some people seem to naturally sense the mood of a room the moment they walk in, while others focus entirely on the logistics of the event? The female brain's default towards social monitoring means that even when at rest, the regions associated with thinking about others remain highly active. This heightened connectivity in the default mode network ensures that social relationships and the nuances of communication stay at the forefront of the cognitive experience. The male brain's tendency to shift between focused systemizing and social engagement provides a different kind of utility, offering a specialized clarity when a system needs to be fixed or a goal needs to be reached. Together, these two neurological strategies ensure that humanity can both build complex structures and maintain the deep emotional bonds that hold a community together. The biological drive to take risks is rooted deeply in the architecture of the ventral striatum, a key component of the brain's reward system. This region acts as a neural hub for processing dopamine, the neurotransmitter associated with motivation, anticipation, and pleasure. In the male brain, the density and sensitivity of dopamine receptors often create a higher threshold for chemical satisfaction. This physiological reality means that certain activities must be more intense, competitive, or uncertain to trigger a significant neurochemical reward. Evolutionary biologists often point to the high-stakes environment of ancestral hunting and territorial defense to explain this divergence in reward processing. For ancient males, the potential for a massive caloric payoff or improved social standing often outweighed the immediate physical dangers of a pursuit. This history has left a permanent footprint on the modern nervous system, manifesting in what researchers sometimes call young male syndrome. During late adolescence and early adulthood, the brain's risk assessment centers are temporarily dampened while the reward circuits are hypersensitized. The influx of testosterone during this period further desensitizes the amygdala to fear and heightens the allure of status-seeking behaviors. While this can lead to dangerous impulsivity, It also historically drove the exploration of new territories and the development of innovative tools. The female brain typically displays a different calibration within these same reward systems, often showing a significantly higher sensitivity to potential punishment or negative consequences. Neuroimaging studies reveal that women often have more active neural pathways in the orbital prefrontal cortex when weighing the risks of a decision. This area of the brain is responsible for evaluating sensory expectations and adjusting behavior based on the likelihood of a bad outcome. This does not imply an absence of risk-taking, but rather a structural preference for calculated safety. The female reward system is often more responsive to consistent, smaller gains rather than the all-or-nothing gambles that appeal to the male dopamine profile. When faced with extreme uncertainty, male neural activity tends to concentrate on the magnitude of the potential prize. This singular focus can lead to a phenomenon where the drive to win overrides the logical signal to retreat, even when the odds of success are dwindling. In contrast, female decision-making processes under stress tend to involve a more robust calculation of probability. The brain filters the situation through a lens of preservation, prioritizing the protection of existing resources and social stability. These structural differences in the ventral striatum and prefrontal cortex ensure a balance of strategies within a human group. A community benefits from having some members who are biologically wired to take the high-stakes risks necessary for growth and others who are wired to perceive and avoid the subtle signs of impending danger. This neurological diversity creates a resilient social fabric where both progress and preservation are given equal weight in the survival of the species. Deep within the primate brain, a specialized cluster of neurons works silently to map out our position within the human landscape. This neural mapping system functions like a social compass, constantly scanning for cues of rank, authority, and influence to determine how we should interact with those around us. In the male brain, this internal architecture often displays an heightened sensitivity to hierarchical structures and clear markers of status. Studies using functional neuroimaging have shown that when men observe a social ladder, their brains tend to activate regions associated with competitive drive and resource acquisition. This biological focus on rank likely stems from ancient evolutionary pressures where a male's position in the hierarchy directly influenced his access to food, safety, and reproductive opportunities. Challenges to this perceived status trigger intense activity in the male amygdala and the hypothalamus, areas responsible for the body's physiological stress response. When a male's rank is threatened, his brain may experience a surge in testosterone that prepares him for a dominance contest, whether that contest is physical, verbal, or purely professional. Interestingly, the male brain often finds a sense of neurochemical security in knowing exactly where everyone stands, as clear hierarchies reduce the unpredictability of social interactions. This preference for structured rank allows for efficient group coordination during high-stakes tasks, where a clear chain of command prevents internal friction. The female brain frequently processes these social dynamics through a different lens, often prioritizing egalitarianism and the maintenance of social consensus over vertical rank. Research indicates that women typically show higher activation in the prefrontal cortex during social interactions, a region involved in understanding the perspectives and needs of others. This horizontal social mapping favors the creation of webs of connection rather than ladders of power, where the primary goal is to maintain group harmony. By focusing on consensus, the female brain ensures that resources and information are shared more fluidly across the community, which historically increased the survival rate of offspring. Regardless of these structural leanings, both sexes share a profound biological vulnerability to social rejection, which the brain processes using the same neural circuitry as physical pain. When we are excluded from a group or lose social standing, the dorsal anterior cingulate cortex fires in a way that is nearly identical to the sensation of a physical injury. This overlap exists because, for our ancestors, being cast out of the tribe was a death sentence, making social pain a critical survival signal. The feeling of hurt feelings is not just a metaphor but a literal physiological event designed to keep us integrated within the safety of the collective. How would you describe your own reaction to a shift in social dynamics? Do you find yourself looking for a clear leader, or do you prefer the group to reach a natural agreement? Status challenges also tend to ignite different neural regions depending on the observer's biology. In many male subjects, a loss of status activates the subcortical regions associated with aggression and autonomic arousal, suggesting a defend the position reflex. Conversely, in many female subjects, similar social stressors more frequently activate the insular cortex, which is deeply involved in processing empathy and internal bodily sensations. This suggests that while a man might perceive a status drop as a challenge to be overcome, a woman might perceive it as a rupture in the social fabric that needs to be mended. These diverse biological strategies ensure that a human group can both compete for excellence and cooperate for stability. The internal rhythm of the human body operates on a clock that is not perfectly set to 24 hours, and this biological timing varies significantly between the sexes. Research published in the Proceedings of the National Academy of Sciences indicates that the female circadian clock often runs about 6 minutes faster than the male clock, creating a subtle but persistent shift in sleep-wake cycles. This slight acceleration means women are more likely to be morning larks, naturally waking up earlier and reaching peak alertness sooner in the day. Men, conversely, often possess a cycle that leans slightly longer, which frequently pushes their peak energy levels into the later evening hours. Such a discrepancy can lead to a phenomenon known as social jet lag, where the body's internal timing is constantly at odds with the external schedule of the modern world. Women frequently report higher rates of insomnia and fragmented sleep, a trend that appears to be rooted in a state of neurological hyperarousal. This heightened sensitivity in the brain's monitoring systems means that the transition from wakefulness to deep sleep is often more precarious for the female mind. While the brain is resting, certain regions involved in emotional processing and environmental monitoring remain more active in women, making them more susceptible to being jolted awake by minor noises or temperature changes. This vigilance likely served an evolutionary purpose in protecting offspring, but in a contemporary setting, it often results in a struggle to maintain a continuous, restorative slumber. How do you find your own energy levels shifting as the sun sets? Do you feel a second wind of creativity, or does your body begin its quiet descent into rest early in the evening? While women struggle with the initiation of sleep, the male brain faces its own distinct structural challenges, most notably a higher prevalence of obstructive sleep apnea. Men are biologically predisposed to carry more adipose tissue around the neck and upper airway, and their pharyngeal anatomy is often narrower and more prone to collapse during the deep relaxation of sleep. When the throat muscles relax, the airway can momentarily close, forcing the brain to trigger a mini-arousal to restart breathing. This cycle can happen dozens of times per hour, preventing the brain from ever reaching the deep, slow-wave stages required for metabolic cleanup. Because of these structural differences, men often experience profound daytime fatigue despite spending a full 8 hours in bed. The specific wiring of the female brain, which utilizes a more distributed and interconnected network of neurons, may actually necessitate a longer recovery period. Because the female cortex tends to engage in more multi-track processing throughout the day, it accumulates a higher sleep debt that can only be cleared through extended periods of rest. Some neuroscientists suggest that because women use more of their brain for complex social and linguistic tasks, they may require approximately 20 minutes more sleep than men to achieve the same level of cognitive restoration. Without this extra time, the prefrontal cortex, the area responsible for executive function, shows a more rapid decline in performance and a heightened sensitivity to stress. Internal temperature regulation acts as another point of divergence, as the female body typically maintains a slightly higher core temperature but colder extremities. This thermal gradient is crucial because the brain requires a drop in core temperature to initiate the production of melatonin and enter deep sleep. Women often find it harder to shed that internal heat, leading to a restlessness that persists until the body can find a thermal equilibrium. Men generally have a higher metabolic rate that generates more heat, yet their skin temperature tends to be more uniform, allowing for a more straightforward cooling process. These biological nuances demonstrate that while sleep is a universal human need, the path to achieving it is uniquely paved by our underlying physiology. The landscape of mental health is not a separate entity from physical biology, but rather represents the visible expression of our underlying neural architecture. These patterns emerge from the specific ways different regions of the brain communicate and how they react to the chemical environment surrounding them. Statistics from the World Health Organization show that women are diagnosed with depression and anxiety at significantly higher rates than men, a phenomenon that has deep roots in the way the female brain processes emotional stimuli. A key component of this susceptibility is the default mode network, a system that becomes active when the mind is at rest or reflecting inward. In the female brain, this network often displays higher levels of connectivity with the emotional centers of the limbic system, leading to a cognitive pattern known as rumination. This process involves the repetitive focusing on the causes and consequences of one's distress, which can inadvertently strengthen the neural pathways associated with low mood. Men's brains typically show a more rapid transition from this internal reflection to the task-positive network, which often shifts the focus from feeling to external action. This structural framework is constantly influenced by a shifting sea of hormones that act as powerful modulators of brain chemistry. Estrogen is far more than a reproductive hormone. It is a critical neurosteroid that directly influences the production and degradation of serotonin, the brain's primary mood stabilizer. When estrogen levels drop sharply, such as during the luteal phase of the menstrual cycle or throughout the transition into menopause, the brain may experience a temporary deficit in serotonin availability. Research published in the journal Science suggests that the male brain is capable of synthesizing serotonin at a rate approximately 52% faster than the female brain. Such a disparity in synthesis speed might explain why men often recover from acute stressors more quickly on a chemical level. Women, however, tend to have a higher density of serotonin receptors in certain regions, which can make the system more sensitive to even minor fluctuations in the neurotransmitters levels. The hippocampus, which is central to memory and mood regulation, possesses a high concentration of receptors for cortisol, the primary stress hormone. Because the female hippocampus is often more densely packed with these receptors, it can become more easily overwhelmed by chronic stress, leading to a persistent state of hypervigilance. The anterior cingulate cortex, often described as the center that processes the physical pain of social rejection, also tends to show higher activity in the female brain during interpersonal conflicts. Individual differences in the GABA system, which provides the brain's natural braking mechanism, further contribute to how anxiety is experienced. Variations in the sensitivity of these calming pathways mean that a level of stimulation that feels exciting to one person might feel overwhelming to another. These biological predispositions do not dictate a person's fate, but they do provide a necessary map for understanding why certain minds are more vulnerable to the heavy clouds of anxiety or the quiet stillness of depression. Knowledge of these internal systems allows for a more compassionate view of the struggles occurring beneath the surface of the skin. Neurodevelopmental conditions frequently reveal a stark divergence in how different brains navigate the world from a very young age. Statistics consistently show that boys are diagnosed with autism spectrum disorder and attention deficit hyperactivity disorder at significantly higher rates than girls. One influential perspective on this is the extreme male brain theory, which suggests that certain neurodevelopmental conditions represent an exaggeration of typically masculine cognitive traits. In this framework, the brain is viewed on a spectrum between systemizing, which is the drive to analyze and build rule-based systems, and empathizing, which is the drive to understand and respond to the emotions of others. While most people possess a balance of both, an extreme tilt toward systemizing can result in a mind that excels at understanding mechanical or mathematical structures but struggles with the unpredictable nuances of social interaction. This imbalance is often reflected in the physical architecture of the brain, where a high degree of local connectivity within specific regions comes at the cost of long-range connections between distant areas. Such a layout allows for intense focus on specific details but makes it difficult to integrate diverse types of information simultaneously. The male brain also appears more vulnerable to certain genetic mutations because of the presence of only one X chromosome. This lack of a backup copy means that if a gene related to brain development on the X chromosome is damaged, there is no second healthy version to compensate for it. This biological reality provides a form of protection for the female brain, which can often bypass the effects of a single genetic error. The dopamine reward system further differentiates these vulnerabilities, particularly regarding substance use and risk-taking. Men are statistically more likely to engage in externalizing behaviors, such as seeking out high-intensity stimuli or substances to satisfy a more demanding dopamine circuit. This drive for a greater chemical hit can lead to higher rates of addiction as the brain attempts to find equilibrium through external means. Interestingly, when conditions like ADHD are present in both sexes, they often wear very different masks. In the male brain, ADHD frequently manifests as physical hyperactivity and impulsive outward behavior, making it highly visible to teachers and parents. In the female brain, the same underlying neurological struggle often manifests as internal restlessness or chronic daydreaming. Because these symptoms are quiet and do not disrupt the environment, they frequently go unnoticed for decades. This leads to a phenomenon where girls develop sophisticated masking strategies, using their generally stronger social intuition to blend in while their internal cognitive load remains heavy. The female brain's tendency to internalize struggle means that what looks like a lack of focus might actually be an exhausted mind trying to keep up with a world not designed for its specific processing style. Understanding these distinct patterns allows us to see that the same biological root can grow into vastly different experiences depending on the structural landscape of the mind. Knowledge of these differences is not about setting limits, but about providing the right tools for every unique architecture to thrive. While average trends offer a window into biological patterns, the reality of any single person is rarely a perfect reflection of a group statistic. This led Professor Daphna Joel and her team at Tel Aviv University to challenge the traditional binary view of the human mind. Their extensive research into brain scans revealed that most individuals do not possess a brain that is consistently male or consistently female across every single region. Instead, the human brain is better described as a mosaic, composed of a unique combination of features that may lean toward one end of the spectrum or the other. This means a single brain might have an amygdala with a volume typically seen in men, while simultaneously possessing a hippocampus with a density more characteristic of women. Such a mix is not an anomaly. It is the standard biological reality for the vast majority of people. If you think of the brain as a complex soundboard with hundreds of different sliders and dials, it becomes easier to visualize this variation. Each slider represents a specific trait, such as verbal fluency, spatial rotation, or emotional processing speed. In this metaphor, there are no male or female soundboards, only unique configurations where some sliders are pushed higher and others are pulled lower. While certain configurations might be more common in one sex, every individual ends up with a custom setting that belongs only to them. This biological collage explains why a person might feel they align with their sex in some cognitive areas while feeling completely different in others. The existence of group averages in neuroscience does not negate the existence of individual diversity, but rather provides a backdrop for it. We can observe that, on average, the female brain has more robust connections between hemispheres, yet an individual woman may have a wiring pattern that looks more like the male average. These overlapping curves in data show that the differences within a group are often much larger than the differences between the two groups. It is this internal variety that allows for the immense range of personalities and talents we see in the human population. When we stop viewing the brain as a choice between two separate buckets, the science of sleep and neurobiology becomes much more nuanced. The mosaic theory suggests that our brains are built to be flexible and adaptable, rather than rigid replicas of a gendered template. This structural diversity ensures that any given population has a wide array of problem-solving styles and emotional temperaments available to it. Nature seems to favor this blending of traits because it creates a more resilient and capable species. Every mind is a distinct work of art, shaped by a prehistoric blueprint but finished with a highly personal touch. Does your own way of thinking feel like a blend of different strengths? Or do you find you align more closely with one specific cognitive style? This sense of internal balance is exactly what the mosaic theory seeks to explain by looking at the brain as a whole. By moving away from a binary model, researchers can more accurately study how hormones, environment, and genetics interact to create a person. This shift in perspective allows us to appreciate the biological complexity of the mind without feeling the need to categorize every trait into a specific box. The human experience is far too rich for a simple two-tone map, and our neural architecture reflects that magnificent variety. Every time a person repeats a specific action or engages in a certain type of thought, the physical structure of the brain begins to reorganize itself through a process known as neuroplasticity. This adaptability means that the neural pathways we use most frequently become more efficient, thicker, and more heavily myelinated over time. If a child is encouraged toward mechanical puzzles, their parietal lobes, the regions governing spatial awareness, develop stronger connections to reflect that constant practice. Cultural expectations often act as a powerful invisible hand in this process, guiding different sexes towards specific activities that reinforce their initial biological predispositions. This creates a feedback loop where a small natural inclination is magnified by years of social reinforcement, making a learned skill eventually look like an innate biological trait. Research has demonstrated that the perceived gaps between male and female cognitive strengths are far more flexible than once believed by the scientific community. One notable study involving mental rotation, a skill where men typically score higher, showed that just 10 hours of playing action video games could nearly eliminate the performance gap for female participants. This suggests that the brain is not a static organ, but a highly responsive machine that builds whatever tools its environment demands. When women are given the same spatial training as men, their neural architecture adapts rapidly, proving that the hardware of the mind is always ready for an upgrade. Similarly, when men engage in roles that require high levels of verbal nuance or emotional tracking, their language centers and empathy circuits show measurable growth and increased connectivity. The famous London taxi driver study provides a perfect scientific lens for understanding how specialized knowledge alters the human head. Researchers discovered that taxi drivers who spent years navigating the complex streets of London developed a significantly larger posterior hippocampus, the area of the brain responsible for spatial memory. This phenomenon is not limited by sex. Any brain, regardless of its baseline, will physically expand and refine itself when pushed to master a difficult task. In the context of gendered skills, this means that many differences we observe in adults may simply be the result of a cognitive London they have been navigating since childhood. If a society encourages one group to focus on navigation and another on verbal communication, the brains of those groups will inevitably begin to look different under a microscope. The adaptability of the human mind is perhaps its most impressive feature, serving as a bridge between our genetic code and our lived experience. While hormones and prenatal development provide a starting point, they do not write the final script for how a person thinks or behaves. Neuroplasticity ensures that a man can develop the delicate verbal precision of a poet just as a woman can master the complex spatial reasoning of a structural engineer. This biological flexibility suggests that our potential is not a fixed quantity determined at birth, but a moving target that shifts with every new hobby, career, or habit we adopt. The human brain is essentially a living record of how we have spent our time. This means that any discussion of biological differences must always account for the transformative power of experience. When we look at the brain, we are not just looking at a male or female template. We are looking at the accumulated history of every problem solved and every word spoken. This incredible resilience allows the mind to transcend its own baseline, constantly rewriting its own connections to meet the challenges of the world. Each individual possesses a unique neural map that is as much a product of their will as it is their biology. The transition into parenthood triggers a biological metamorphosis that is among the most profound shifts a human brain can undergo. During pregnancy, the female brain experiences a significant reduction in gray matter volume in regions tied to social cognition. Far from being a loss of intelligence, this process represents a highly specialized form of synaptic pruning, stripping away less used connections to make the neural architecture more efficient. Researchers have noted that these structural changes are so distinct they can be used to identify whether a woman has ever been pregnant through a brain scan alone. This remodeling focuses on the medial prefrontal and posterior cingulate cortex, areas responsible for empathy and the ability to perceive the mental states of others. Nature ensures these adaptations remain in place for years, effectively hardwiring the mother to the survival needs of her offspring. While these changes are driven by the hormonal surges of pregnancy, the male brain is equally capable of significant biological reorganization through the act of caregiving itself. When a man becomes a father, his testosterone levels often experience a measurable decline, a shift that is thought to reduce aggression and increase his sensitivity to the infant. Simultaneously, his levels of oxytocin and vasopressin rise, hormones that facilitate bonding and protective behaviors. This chemical recalibration is particularly pronounced in men who spend the most time in direct physical contact with their children. The sound of an infant's cry acts as a powerful neurological trigger, activating the amygdala and the hypothalamus with startling speed. In a parent's brain, these regions bypass the slower, more analytical parts of the cortex to initiate an immediate caregiving response. This heightened sensitivity is not limited to biological mothers. It develops in any adult who takes on the role of a primary caregiver. Brain imaging studies have shown that when fathers are the primary source of care, their amygdala activity matches that of mothers, suggesting that the brain's parenting network is a universal human capacity. This neural sensitization creates a permanent footprint, altering how the brain processes emotional information for the rest of an individual's life. Progesterone and estrogen levels during the postpartum period further enhance this plasticity, allowing the brain to rapidly learn the nuances of a specific baby's nonverbal cues. Even the way a parent perceives smells or recognizes faces is subtly altered to favor the detection of their child's unique signature. Such biological shifts prove that the brain is not a static organ but a responsive system that adapts to the survival requirements of the next generation. Whether through the massive hormonal shifts of pregnancy or the steady, daily interaction of a father, the brain prioritizes the development of a social radar specifically tuned to the child. This shared neurological foundation suggests that the capacity for deep, nurturing care is a fundamental part of the human biological toolkit, transcending sex to ensure the protection of the vulnerable. As the years advance, the human brain undergoes a significant metabolic transition that differs sharply between the sexes. In the female brain, the onset of menopause marks a sudden and dramatic withdrawal of estrogen, a hormone that serves as a primary fuel regulator for neural tissue. Estrogen acts as a master key, helping the brain's mitochondria effectively convert glucose into cellular energy. When these levels plummet, the brain often enters what researchers describe as a temporary energy crisis. This biological shift frequently manifests as a period of brain fog, characterized by lapses in short-term memory and a decreased ability to concentrate. Imaging studies from institutions like Weill Cornell Medicine show that the brain's glucose metabolism can drop by up to 25% during this transition. This metabolic dip is a structural reorganization rather than a permanent loss of function. Over a period of several years, the female brain typically stabilizes, adapting its chemistry to utilize alternative energy sources more efficiently. During this period of recalibration, many women find that their cognitive clarity returns as the neural architecture settles into a new, post-reproductive equilibrium. The male experience of aging follows a markedly different trajectory, often referred to as andropause. Unlike the sudden hormonal cliff of menopause, testosterone levels in men tend to decline in a slow, linear fashion, dropping by roughly 1% each year after the age of 30. This gradual reduction means the male brain rarely faces the acute metabolic shock seen in females. Because the change is so incremental, the neurological impacts on mood and cognitive speed are often subtle and can span several decades. While this provides a more stable transition, the long-term decline in testosterone is eventually linked to a thinning of the cortex and a gradual reduction in muscle-to-fat ratios that can affect overall brain health. Aging acts as a great equalizer, slowly narrowing the cognitive gaps that were more prominent during the peak reproductive years. As sex hormone levels diminish in both men and women, the brain moves toward what some neuroscientists call a more unisex state. Differences in verbal fluency and spatial rotation, which are often distinct in younger adults, begin to blur as the underlying hormonal drivers of those specializations recede. This convergence suggests that the high-contrast cognitive profiles of youth are temporary adaptations rather than fixed, lifelong identities. Emotional processing also shifts during these later stages of life, often leading to a phenomenon known as the positivity effect. Both men and women show a trend toward better emotional regulation, with the amygdala becoming less reactive to negative stimuli and more focused on rewarding, peaceful experiences. For many men, the decline in testosterone can lead to a softening of competitive drives, often resulting in increased nurturing behaviors and social connectivity. Conversely, the shift in the estrogen-to-testosterone ratio in aging women can sometimes manifest as a newfound sense of assertiveness and independence. This period of life represents a profound neurological transition where the focus moves from the intense biological demands of reproduction to a more reflective and stable state of consciousness. These changes ensure that the brain remains a flexible organ, capable of finding new ways to maintain wisdom and social utility well into the twilight years. Statistical data from the World Health Organization reveals that women are diagnosed with Alzheimer's disease at nearly twice the rate of men, a discrepancy that extends beyond the simple fact that women generally live longer. Recent neurological research suggests that the female brain undergoes a unique metabolic shift during the transition into the postmenopausal years. Estrogen plays a vital role in the brain by promoting glucose metabolism, which essentially provides the fuel necessary for neurons to function and repair themselves. When estrogen levels drop significantly, the brain may experience a period of metabolic fuel failure, forcing it to find alternative energy sources, which can sometimes lead to the accumulation of amyloid beta plaques. This transition period is now viewed by many researchers as a critical window where the neurological foundations for later cognitive decline might be laid. The male brain faces its own distinct set of challenges related to aging and longevity, often centered on the health of the circulatory system. Men are biologically more susceptible to vascular issues and microstrokes, which can lead to a condition known as vascular dementia. While the female brain is more prone to the protein folding issues of Alzheimer's, the male brain is often more vulnerable to the physical degradation of the small blood vessels that supply the white matter. High blood pressure and arterial stiffness in men can cause silent, infinitesimal damages to the brain's wiring over decades. These structural interruptions can eventually manifest as a decline in processing speed or executive function, often appearing earlier in the lifespan than the typical onset of Alzheimer's symptoms. Have you ever noticed how some people seem to stay incredibly sharp even into their 90s despite having physical markers of brain aging? This phenomenon is often explained by the cognitive reserve hypothesis, which suggests that the brain's ability to improvise and find alternate pathways can buffer against physical damage. Cognitive reserve is built through years of education, complex social interactions, and the mastery of multiple skills, creating a dense web of neural connections. If one pathway is blocked by disease or age, a brain with high reserve can reroute information through an existing detour. Research indicates that while women often have higher verbal cognitive reserves, men may sometimes possess a larger brain volume that provides a different kind of structural redundancy. Longevity itself is a complex biological puzzle where the female body consistently demonstrates a survival advantage, yet the brain must work harder to maintain itself across those extra years. The female immune system is generally more robust, which helps in fighting off infections that can accelerate brain aging, but this same hyperactivity can sometimes lead to chronic neuroinflammation. In contrast, the male brain tends to age more linearly, but it lacks some of the protective inflammatory modulations that estrogen provides earlier in life. In extreme old age, the brain survives by pruning away the non-essential, focusing its remaining metabolic resources on the core regions responsible for personality and long-term memory. This persistence of the self, even as the physical architecture thins, showcases the incredible resilience of human biology as it navigates the final stages of the life cycle. The horizon of neuroscience in 2026 is defined by a move away from generalized group averages toward a granular understanding of the individual's biological life path. Researchers are increasingly focusing on the concept of precision neurology, where the specific hormonal and genetic makeup of a person determines the approach to their mental health and cognitive care. This perspective recognizes that a male brain and a female brain may arrive at the same cognitive destination using entirely different neural highways. Personalized medicine is currently revolutionizing how we treat conditions like depression or chronic fatigue by acknowledging that the brain's chemical receptivity is often sex-dependent. Pharmacologists now observe that certain neuroprotective drugs are metabolized more efficiently in the presence of estrogen, while others require the specific metabolic environment of the male body to reach their full potential. This nuanced approach ensures that the biology of the patient is no longer treated as a secondary variable in the pursuit of healing. The gut-brain axis has emerged as a critical field where sex-specific differences are surprisingly pronounced. Science has found that the microbiome, the vast community of microbes in the intestinal tract, interacts with the brain's stress response system in ways that are heavily influenced by circulating sex hormones. In the female brain, the gut-brain link may be more sensitive to fluctuations in the reproductive cycle, whereas in the male brain, it often shows a more consistent, linear relationship with immune function. Artificial intelligence is playing a pivotal role in this exploration by processing thousands of functional MRI scans to detect patterns that are nearly invisible to human observation. Recent studies from institutions like Stanford Medicine have demonstrated that machine learning can distinguish between male and female brain activity patterns with over 90% accuracy, focusing on the specific hotspots of connectivity that define our unique cognitive styles. These digital tools are not seeking to create divisions, but rather to map the exquisite complexity of how human biology organizes information. The modern scientific consensus has firmly shifted away from the binary competition of better or worse toward a sophisticated model of complementary strategies. We are beginning to see the brain not as a rigid structure, but as a flexible organ that adapts its hardware to meet the demands of its environment and its unique internal chemistry. This evolving map of the mind suggests that diversity in neural architecture is one of the most significant assets of the human species, providing a wide array of cognitive tools for survival and innovation. The intricate tapestry of human evolution suggests that the divergence in neural architecture was never a competition, but a sophisticated division of cognitive labor. When ancestral groups navigated the challenges of the Pleistocene, the survival of the collective depended on a diverse toolkit of mental strategies. The male brain's historical leaning towards spatial navigation and systemic logic provided the necessary precision for tracking resources and defending the perimeter. Simultaneously, the female brain's heightened sensitivity to social cues and verbal nuance ensured the cohesion of the group and the survival of the most vulnerable members. This biological specialization meant that a community was always greater than the sum of its individual parts. Resilience in a species often stems from its ability to solve problems from multiple angles at once. A group composed entirely of systemizers might excel at building tools and mapping terrain, but could struggle with the subtle emotional undercurrents that maintain tribal peace. Conversely, a group focused solely on empathy might maintain perfect harmony but lack the detached analytical drive needed to overcome mechanical or structural obstacles. By weaving these two distinct cognitive styles into the fabric of a single species, nature created a balanced system of checks and balances. The overlap between these two worlds is where the most profound human achievements are born, blending the how of the physical world with the why of the social one. The biology of partnership functions much like a masterfully composed piece of music, where different frequencies and rhythms combine to create a resonance that a single note could never achieve. Science suggests that the most successful human units, whether they are hunting parties of the past or modern research teams, thrive when they integrate these varied neurological strengths. The subtle differences in how we process stress, remember details, or perceive color are not barriers to understanding. They are the very assets that allowed our ancestors to colonize every corner of the globe. In the quiet of the night, it is helpful to view these biological variations as a form of deep-seated cooperation that has been written into our DNA over millions of years. This complementarity allows humanity to be both fierce and nurturing, analytical and intuitive, all at the same time. We are a species defined not by our uniformity, but by our ability to bridge the gap between different ways of seeing the world. This ancient partnership remains the bedrock of our shared journey through time. The journey through these myriad biological landscapes reveals a map of human nature that is far more intricate than any simple binary could ever suggest. We have traveled from the microscopic surges of prenatal hormones to the vast architectural networks that define how we perceive a sunset or navigate a crowded room. Each individual carries a unique mosaic of these traits, blending the analytical precision of one system with the empathetic resonance of another. This diversity of thought and feeling is not a collection of isolated islands, but a single, vast continent of human experience. Our brains are living records of 1,000,000 years of survival, adaptation, and profound cooperation. Plasticity remains the most hopeful chapter in this entire scientific story, reminding us that no structure is ever truly fixed in stone. Every conversation we have and every new skill we master physically reshapes the folds of the cortex, blurring the lines between what we were born with and what we have become. This constant state of flux means that while biology provides the starting point, our experiences write the rest of the narrative. The human mind is a master of adaptation, capable of bridging gaps and finding new ways to solve the ancient problems of connection and understanding. We are witnesses to a biological miracle that repeats itself with every heartbeat and every firing neuron. Science for sleep thrives on this sense of quiet wonder, where the complexity of the world becomes a source of peace rather than a cause for stress. Understanding the mechanical how of our existence often brings a deeper appreciation for the why that guides our daily lives. As the quiet of the night settles in, the heavy lifting of the day's cognition gives way to the restorative work of the resting mind. This transition is a universal human rhythm, shared by every person regardless of their specific neural wiring or hormonal profile. It is the moment when the biological machinery of the self finally pauses to integrate the lessons of the waking world. The shared experience of consciousness remains the ultimate frontier, a place where all our structural differences eventually converge into a single stream of awareness. If this exploration of the biological mind has brought you a sense of clarity or calm, consider subscribing to join our future journeys into the wonders of the natural world. Every fact we have uncovered serves as a reminder that we are part of something much larger and more ancient than ourselves. Sleep now with the knowledge that your brain is a masterpiece of evolution, working tirelessly even in silence to keep the flame of your curiosity alive. The world remains full of mysteries waiting for the morning light, but for now, the only task left is to simply exist within the quiet pulse of the universe.